Okay, so this was a project that was created by Quincy Bock for SPU 20. We had asked the students in their final projects to create something that we could use in a didactic way to try to explain some concept from the course, and what Quincy decided to present was an interactive sculpture that would illustrate the famous X-ray photograph taken by Rosalind Franklin that was used by Watson and Crick in proposing the structure of DNA. So this is the photograph, and what Quincy has done here is she's made a slide of the photograph. So this is the actual X-ray photograph called Photograph 51, taken by Rosalind Franklin, and Quincy has put this photograph on a slide with a small clay model of the structure of DNA on the top. So this is, in a sense, if you have the entire structure of DNA, this is the image that you'll see. Now, this photograph is a famous photograph in the history of science, but it's never been, I think, very clear, especially to students in approaching this who don't understand X-ray crystallography, exactly what about this image shows the structure of DNA. So Quincy set out to try to explain that using this sculpture. It's essentially a slide projector. So she built this entire wood box, beautifully constructed, has a light in the back. And what she did was she created individual slides that would highlight certain elements of this structure. So the first one is this slide. And you see here these diagonal X patterns. And at the top, she's put a helix. So there's a single helix here that's illustrated in green. And when you slide this helix in to the slide, it highlights on the image the fact that there's this X cross pattern in the actual photograph. And this illustrates that if you have a helix, as is indicated by this sort of clay icon, when you have a helix, you get an X-ray crystal structure that has this diagonal cross pattern. But of course, DNA isn't simply a single helix. DNA is a double helix. And it turns out that if you have a double helix, which she's represented here by two helices on this image, if you have a double helix, you actually get these band patterns in that diagonal structure. And so here's the double helix pattern showing the bands, the individual bands there. And she's highlighted one aspect of this that's particularly interesting, which is that there's a band missing at this fourth position on the X in each direction. So if you put together the fact that it's a double helix and the fact that it is helical in general, you can combine these two slides and you highlight the helical aspects of this structure. So you highlight the X pattern and you highlight the fact that it has this banding. So that explains some of the more important features of this structure. The other aspects of the structure, which are more subtle, are the fact that there are these blank diamond spaces in the structure. And this is an illustration of the fact that there are individual groups, repeating units, arranged along the DNA backbone. So here we have an illustration of the sugar phosphate backbone. And if you have these sugar phosphate backbone units, you'll get this diamond pattern illustrated here. And this slide highlights that diamond pattern. And one of the crucial elements of the structure is not just the fact that there's this diamond pattern, but that in fact the diamond pattern has a slight variation in intensity that is difficult to see, I think, here, but that the diamonds on the sides are slightly darker than the diamonds in the middle. And what that says, which is illustrated by this slide, is that this sugar phosphate repeating backbone is actually on the outside of the helix. So the fact that the sugars and phosphate groups, the linking groups, are on the outside of the helix means that the diamond shapes here will be slightly darker, the ones on the horizontal axis, slightly darker than the ones on the vertical axis. And that's illustrated by this slide that highlights the fact that you have darker patterns on the two diamonds on the side and lighter patterns on the top and bottom. And that fact that the sugars and phosphates were on the outside of the helix was one of the crucial components of the structure of DNA, and in fact was a significant difference between the structure that Watson and Crick proposed compared with the previous structure that was proposed by Pauling that turned out to be incorrect. And if I put together all of these slides, then they combine to show the entire pattern illustrating the diagonal X, the banding structure, the diagonal X showing a single helix, so there's the diagonal X showing a single helix, the banding structure, which shows that it's a double helix, the fact that you have these diagonal patterns, diagonal spaces on the top and bottom and left and right, which shows this repeating pattern of the sugar phosphate backbone, and the fact that there's a slight intensity difference, darker patterns on the sides, that shows that the sugar phosphate backbone's on the outside. And when you put it all together, you get a pedagogical illustration of this famous photograph, photograph 51 from Rosalind Franklin. And this is such a lovely illustration of this image, an explanation of how this image was important to Watson and Crick in their discovery of the structure of DNA. And I certainly intend to use this in our discussion of the structure of DNA in future courses, because it explains it in a way that really is far better than we had explained it ourselves in the course. And I think Quincy, who is a VES concentrator, who did a beautiful job of constructing this and really went into depth into thinking about how this could illustrate the image here, I think is a truly remarkable example of what we can do in a gen ed course.